Hi class. So we're going to get started with chapter 25, which is on metabolism and nutrition. So metabolism is something we covered a lot in my upper division classes when I took a bunch of biochem. Nutrition, on the other hand, is kind of my weaker areas. They did cover it in medical school, but literally for only like a couple of weeks, which is surprising because they really should pay more attention to that because it's such a major impact on our overall health. But let's talk about metabolism first. So metabolism is the sum of all chemical reactions. And if you remember from Bio 430, it's really one of the major characteristics of life. And our focus is really on carbohydrate metabolism. But in the next lecture, we'll talk also about how lipids are broken down and built up, and even proteins. We'll talk about the different fed states that happen. You could either be in this fasting state or the fed state. The fed state is sometimes referred to as the absorptive state because you're absorbing things, and the fasting state is sometimes referred to as post-absorptive. All right, when we think about metabolism, really the reactions can lead to a buildup of chemicals or a synthesis, and that's referred to as anabolism. And then when things are getting broken down, the opposite of anabolism is catabolism. And one way to remember that is just think about catabolism. Sounds like catastrophe, right? When there's some kind of catastrophe, things are getting broken down, buildings destroyed and things like that. Okay, so these are buildup reactions. These are breakdown reactions. Typically from chemistry, which of these tends to release energy? Yep, so catabolism tends to be energy releasing versus anabolism, energy typically is required. So whenever I just indicate big E, that refers to energy. We have been talking about digestion, and an example of a reaction for digestion for breakdown stuff, that's referred to as hydrolysis. Remember, lysis is to cut, hydro means it's involving water, and this is basically what is happening during digestion. On the flip side, the opposite of hydrolysis, when you're synthesizing things and when you're putting large molecules together, the opposite is referred to as dehydration synthesis. And their names are very useful because hydrolysis is using water, and that water eventually will be part of your reactants, whereas in dehydration synthesis, the water is going to be produced at the end. So when something's dehydrated, water is getting taken out. The other name for these reactions are sometimes called condensation reactions. Now, you can compare anabolism and catabolism by also looking at various processes. And these tend to be very involved biochemical pathways. The main biochemical pathway, when you think about glucose, is this process called cellular respiration. Involved process, probably about 50 steps, and I knew every step at one point when you had to memorize all this in biochem. We're going to distill this down to about 4 main steps. So there's going to be 4 phases that we will focus on. And then the opposite, which is equally as complex, is the one only done by plants. Plants can do both of these, but plants can only do the process known as photosynthesis. Obviously, we don't have time to talk about that if you were to take more cell molecular classes, you would get into that. So let's talk about cellular respiration. Cellular respiration is this universal process, especially if the cells are eukaryotic. That means that they have a nucleus. And again, this process is very complicated, but it can be summarized by an overall equation. And that equation you should commit to memory. C6H12O6 plus 6 oxygens will give you 6 waters, 6 CO2s, and then energy. That energy, the majority of it, will actually be lost as heat, about 60%. And then there's remaining energy that could be harvested to actually use cellular, to make cellular energy, and that cellular energy is ATP. So that's remaining 40%. And when I was taking, early biochem classes, we used to say probably about 36 ATPs are produced. And now the numbers are probably a little bit less efficient than that. So anywhere between 32, 34, probably around 32 ATPs, roughly. Now, there is a major organelle that does cellular respiration. And if you remember from 430, a lot of these processes will happen in the mitochondria. Now, ultimately, our energy, a lot of our energy comes from glucose. We can get energy, of course, from fats and proteins. We'll get into that later, especially on the next lecture. But glucose is this molecule right here. And when that glucose is broken down, you need to break it down in many, many steps. That's why this is such a complicated process. It's about 50 steps. If it's done slowly like that, you can actually capture more ATPs. You can take glucose and burn it, right? You can heat it high enough so that it releases heat, it can sparkle, etc. But that's very inefficient. You're going to release all the energy at one time. So to slowly break down and disassemble the glucose, it will utilize a bunch of energy transfer mechanisms. So basically, the energy from glucose will get passed on like a hot potato to other chemicals, and then that energy eventually will be used to make ATP. Okay, so one energy electron transfer mechanism are these redox reactions. So remember from Gen Chem, there are these redox reactions where something is reduced and then something is oxidized. Okay, remember there's this thing called oil rig, right? Oxidation is loss of that electron, and then reduction is gain. So basically it is electron transfer. And these electrons really contain energy. It turns out in biological systems, oftentimes the electrons can be transferred within a hydrogen atom. So the electron transfer can happen as electrons or electrons within a hydrogen atom. There are some electrons that have more energy. So for example, the electrons in glucose have much more energy, whereas the electrons in stuff like water have very low energy. So we'll get into energy transfer. We're talking about that right now. The second type of ways that energy can be transferred is by transferring a phosphate. Phosphate transfer. If you remember from Bio 430, you have this chemical called adenosine triphosphate, ATP, However, that last bond is very unstable. It wants to release that phosphate. And when it does, you now end up making a DP, and then now you have that released phosphate. Remember, this is catabolic, right? Things are getting broken down. Catabolic reactions release energy. So this is the phosphate group. In that process, energy is released. And now, after taking all these classes, and especially after doing, 1.5 semesters of A&P, you can think where some of this energy is being utilized, right? To move a cilia, to power a pump, to move a flagella, muscle contraction, secretion. All that stuff is requiring energy. All right, so that is phosphate transfer. Now, when you think of this process going the other way, when we're trying to make ATP, one of the fundamental rules in nature is energy is neither created or destroyed. It's basically transferred, right? So you got this ADP. How are you going to remake ATP? Now you have to add energy, right? Going forward is going to be when energy is released, but how do you get more ATPs for biological processes? And that energy will come from food. If we were green and we had chloroplasts, we would be able to sit out in the sun and get food from solar energy, but we can't do that. Unfortunately, that'd be awesome if they designed a cow or a person that was green, because then they could feed off solar energy. But we're not quite there yet. Okay. All right. Third way. It's an interesting one, but concentration gradients. Concentration gradients are really a form of stored energy. That's how batteries work. Basically, there's concentrations of ions that are separated across membranes in a battery, and when that concentration goes from high to low, energy is released to power your drone or whatever toy you have, right? Okay, so concentration gradients, when you go from high to low, you actually will release energy. Versus if you go from a low concentration to a high concentration, energy is required. All right, so those are the main ways that energy will get transferred on its way to ATP. And on this diagram, it just basically rehashes what we talked about. To make ATP, you got to use food molecules, sugars, proteins, triglycerides, okay? And then that ATP can be hydrolyzed. That hydrolysis is going to happen to then release the energy, and that energy can be used to, for cellular processes. Here's the redox reactions we talked about. Something will gain an electron, something will lose an electron. So this is what I was talking about. In Gen Chem, maybe we talked about the electron having, as a electron by itself, but sometimes it is basically you have your electrons within the hydrogen atom. And we'll talk about these chemicals in a moment. All right, so here's the deal. When we talk about cellular respiration, we want to think about, again, it's about energy. And if we had an energy diagram, and in this energy diagram, this had up here had high energy, and down here there was low energy, it would make sense that glucose is going to be way up here. It has high energy. We're going to take that glucose and slowly break it up. And we're going to do it in a step-by-step fashion. So it's going to be 1, 2, 3, and a final step down here. What has low energy are going to be things like water and CO2. So there's going to be 4 main steps that we'll talk about. And in that process, we're going to produce ATP. Now, there are some intermediates here. You may sometimes have to have something carry those electrons. So there are things that we will just call acceptors. They can accept, acceptor, or they can also transfer electrons. So again, pretend we're playing hot potato with the electrons off of glucose. What will capture these electrons? There are chemicals called coenzymes. And it turns out you actually need these coenzymes. You get them from your diet. So one of them is called NAD. And NAD actually comes from a B vitamin called niacin. NAD, some of you guys may be thinking, oh, what does NAD stand for? It's for nicotinamide adenine dinucleotide. You guys can call it NAD. Usually it actually has a plus charge. It can grab a couple of electrons, but these couple of electrons will be packaged into two hydrogens. Remember, each hydrogen atom has one proton and typically one electron. So if you have two hydrogen atoms, you're going to have two electrons bundled in there, right? So these two hydrogen atoms, when they combine with NAD+, you're going to end up making NADH and H+. Bottom line, NAD+ is like a taxi cab for electrons. Okay? They're going to transfer these electrons, hold on to the energy, and eventually these energy chips, like poker chips, will be cashed in to make ATP. There is another one that comes from something called riboflavin. And that riboflavin is used to make something called FAD. Flavin adenine dinucleotide, you guys can call it FAD. Just make a little side note here. Both of these chemicals are examples of B vitamins. B vitamins are critical for energetic metabolism, and that's why a lot of these energy drinks that some of you guys may drink to survive, working crazy jobs, 2 jobs, whatever, they have a lot of B vitamins in them. FAD has a similar role. It can bind to 2 hydrogen atoms. And, you know, if niacin is the taxi, this will be like the Uber, right? After it holds on to those 2 hydrogen atoms, it will now be called FADH2. Another acceptor of electrons, and it can transfer them, we'll talk about cytochromes. They're in the mitochondria. They have an iron core. And they can bind electrons and then transfer them. Lastly, the final electron acceptor. Does anybody remember the most electronegative atom in the periodic table? Remember, it was all the way in the corner. It was fluorine, right? And right next to fluorine is oxygen, right? Oxygen is super electronegative. And remember what electronegative just meant, that it had a strong affinity for electrons. You're like, oh my gosh, there's that word affinity again. Remember, affinity just meant attraction, okay? So oxygen is very electronegative, has a high affinity, high attraction for electrons. So this guy has a lot of electronegativity. It wants electrons, so it's even going to accept the low energy electrons. It's kind of like the garbage man, right? It'll finally do the final pickup of those electrons and then carry them out. Okay? All right. So we talked about these redox reactions. And we'll get into now glucose. Glucose, as it states there, is a major fuel source. Your body is pretty good at using fats when needed too. So, we can convert other things into glucose. So it can be used for energy. Here's that glucose molecule. You don't have to memorize any of that. Now, before we get into the actual steps of cellular respiration, let's talk about glucose getting into cells. So how does glucose enter cells? Now, there are, of course, different transport processes, but a major way that glucose gets in is by using a transporter, and usually it will require some kind of transport protein. One type is called secondary active transport. This is a bizarre one. We'll get back to this later, but this is used more by places like the kidney. And actually, we talked about this before. Some of the GI tract enterocytes in the small intestine use secondary active transport. Really, remember that's a weird one because this does use ATP. And this can go from a low concentration to a high concentration. This is how the body prevents loss of glucose in the GI tract or in the urinary tract. In these areas of your body, your cells are like glucose hogs. They're going to try to hoard as much as possible, and so they have to use a secondary active transport. Now let's focus on the majority. What about the rest of the body? The rest of the body is going to use facilitated diffusion. And for facilitated diffusion to work, you need to have a concentration gradient, and you'll also need a transport protein. Now, there's a lot of your cells in your body that, for them to do this, they are actually insulin dependent. So it's like, okay, you got a concentration gradient, but you need to have like a gatekeeper, and that gatekeeper is insulin, okay? So insulin, in this case, causes a GLUT, which is a glucose transporter, to be expressed. Really big concept, right? Because it ties into diabetes and just normal day-to-day glucose going into your cells. Now, what happens here then is, let's kind of the best way to understand this is pretend that this is a cell, and the cell basically has receptors for insulin. Prepackaged, within the cell are these transport proteins. Let's call these the GLUT. When insulin is present, what will happen there is on the cell, let's say that the insulin receptors are now bound to insulin. Let's say the insulin are the dots here. So this kind of ties back to the first unit. This is almost like the first messenger, right? There's going to be some signals that will cause now changes within the cell, and those changes actually cause these transporters to be expressed on the surface. They transport glucose. These are those glucose transporters, and now they can bind. And as long as you have a concentration gradient, meaning that there's more glucose out here than in here, these glucose molecules can now enter the cell and go inside the cell, right? That, remember, facilitated diffusion uses some kind of transport protein, and you have to have a concentration gradient. So the key here is they are insulin dependent. Got to have insulin around for the glucose to get in. There's also, again, where does this happen? This is most cells of the body. Okay. This actually also includes the satiety center. Oh, I did a terrible job there. So satiety center. This is in the hypothalamus of the brain. So imagine that, right? If you have insulin issues and the insulin's not expressed or being secreted as a hormone, these satiety centers are not going to turn off, right? So people are going to stay hungry. You have to allow the glucose to get in, and then eventually you'll feel satiated or full, right? This is a process, right? So this is the reason why, if you eat slower, if you're like at a buffet or something like that, right? You eat a bunch of food, give yourself like, 10 minutes, you're probably going to feel super full. But we oftentimes don't allow ourselves to get, that feeling of satiety yet because the food is so amazing, okay? All right. Now, are there some cells that are insulin independent? For sure. So this is insulin independent using facilitated diffusion. So for these cells, they're basically expressing the glucose transporter all the time. If glucose is high out here, it'll automatically go into the cell. They don't need insulin to act like a key to open up the gate. So are there some examples of this? Sure, red blood cells, most neurons actually, and some hepatocytes. Both of these, notice this is the majority of the cells, right? Remember if we went back here, we have the secondary active transport where you don't need a concentration gradient, right? Because it uses ATP. For the great majority of your body, it's using some form of facilitated diffusion. So kind of a key point for both of these is you have to have a concentration gradient. Normally our blood glucose is somewhere between 70 to 140 milligrams per deciliter. You have to have at least some glucose in the blood so that they can enter the cells via facilitated diffusion. So you're going to be 70 more when you're in the fasting state versus 140 in the fed state. Does that make sense, right? Got to have some glucose in there. Once you start creeping towards 70 and below, this is when you can become very irritable, hangry. You know what I'm talking about, right? Okay. All right. Now glucose, let's say, has gotten into the cells. It's using some form of facilitated diffusion. So here's some examples. We just discussed that. Here's where we're going now. Let's talk about these overall steps to eventually make ATP. All right, so let's just list the reactions so far, or the reactions that we're going to get into. So this is cellular respiration. And these are really sets of reactions, okay? So first one is glycolysis. This is where you will split the glucose. Lysis is to cut, right? 2 is called the transition, or the Krebs prep, because this will allow for the next step, or allow these chemicals to feed into the next step. It's called the Krebs cycle. Sometimes it's called the citric acid cycle. And then the final one, where you cash in all your chips and make the highest number of ATPs, is the ETC. That is called the electron transport chain. All right, let's go for it. So glycolysis, it's called the Embden-Meyerhof pathway. It's about 10 steps. We'll learn one of those steps, okay? This is a universal pathway. So complex cells do this process. Bacteria use this as well. This is going to happen first, or actually the whole process is happening in the cytoplasm. Again, it's about 10 steps. And one step that I want to get into a little bit of detail, because it makes sense, okay, is step 1. You're actually going to take the glucose and add a phosphate to it. What's going to have phosphate? ATP. So isn't that weird? You're actually spending energy to eventually make more energy, right? It's like investing, right? Put some money in, get some more money out. And hopefully you didn't do that yesterday, because the market, I guess, or about a week ago, the market crashed hard, right? So glucose 6, phosphate is what you make. So why do this? Well, one thing is that it makes glucose more reactive. It destabilizes the glucose. You're kind of priming it. You're getting it ready to get broken down, okay? And then the other part is, if you look here, this is now glucose 6-phosphate, and then the transporters here that we were referring to were glucose transporters, right? GLUT. They're not glucose 6-phosphate transporters. So once you add a phosphate onto the glucose molecule when it's trapped in the cell, or when it's in the cell, you're now going to trap it, because you're still going to maintain really the concentration gradient. This is still going to allow glucose to go in, but because this is a different molecule, it can't go the other way. These glucose transporters can actually work both ways like enzymes, but if it's now glucose 6-phosphate, it cannot leak out of the cell. So it's really interesting. So the other thing it does basically is it helps to trap that glucose into the cell. All right. So you guys probably did this in a lot more detail in classes like micro, or if you've taken a biochem class, but I'd like you to focus on just basically what goes in and what comes out. So we're going to turn glycolysis into a black box. And if my biochem teachers knew that I was doing this, they'd probably be in like massive protests. Some of them are maybe dead now. All right, so glucose goes in. It has 6 carbons. Also, 2 NAD pluses come in. Remember, NAD plus is like a electron carrier, high energy electron carrier. 2 ADPs go in, plus phosphate. After these 10 steps take place, you're now going to have 2 pyruvic acids. These are 3 carbon each, so 2 times 3 gives you back your original 6 carbon glucose. You will make a couple of high energy chemicals, as well as making a couple of ATPs. All right, so that's basically it. If I said, oh, what are the net products of glycolysis? It would be the 2, 2, and 2, right? 2 pyruvic acids, 2 NADHs. You do make a couple of ATPs. All right. Now, NADH, that's going to contain energy. We'll cache that in later. You do make a couple of ATPs, right? Now, it turns out that this chemical right here, NAD+, there's a limited supply of those. Now, if you look over here, you make the NADH. This chemical will eventually go back to NAD+ when these hydrogen atoms are released. And that's going to be released in the final phase, which is the electron transport. That last step, it turns out, requires oxygen for that to work. So, is there a possibility that you can make more of these so that you can continue glycolysis when a person does not have enough oxygen? So in other words, are there tissues in your body that can make ATP without oxygen? There is. And really the main example of that is skeletal muscle. Remember, there are these fast oxidative fibers, fast glycolytic fibers. And what they'll do, the reason why they're called glycolytic, because it ties back to glycolysis. Skeletal muscle, especially the fast glycolytic ones, have a very important enzyme. And the chemical reaction that they will do is they're going to take this pyruvic acid that's at the very end, and they will convert it into lactic acid. Remember, the fast glycolytic is for high intensity activities where there's going to be a lot of lactic acid produced. Now, in that process, it will use an enzyme that is unique to skeletal muscle, and it's called lactate dehydrogenase. We know it's an enzyme because of the ASE, and what that enzyme will do is that it will take that NAD plus and basically remake it, okay? Because the NADH, without oxygen, the NADH is going to build and build and build. Skeletal muscle has this ability because of this enzyme. It can take the NADH and remake NAD plus. That NAD plus can now feed back into glycolysis. And that's the reason why skeletal muscle can continue to do glycolysis, because other tissues, they would not be able to regenerate that NAD+. They'd have to go through all of cellular respiration to remake NAD+. But skeletal muscle can say, well, no, I'm going to go around the other processes and make NAD+, so that we can burn more glucose and make a couple of ATPs. So you may have to re-listen to that section. But the bottom line, skeletal muscle, they have a unique enzyme. They can do a process called, this whole process is called lactic acid fermentation. lactic acid fermentation is basically saying, okay, the last thing that will accept the hydrogens is not going to be oxygen, but it's going to be some kind of organic molecule, and that organic molecule is lactic acid. We have taken the hydrogens, we've pulled them off, we have dehydrogenated, right? Dehydrogenated is what I should have said. Taken the hydrogens off, now you make the NAD+, and then you can keep doing this. Now, can you do this forever for your skeletal muscle? No, because what does lactic acid do eventually? It'll build up, and that'll lead to fatigue. Okay? All right, so that's a special tweak that skeletal muscle can do, and no other tissue can do. Alrighty, now, to talk about the other steps, we got to get into the mitochondria. We'll get into the structure a little bit. This area here is called the matrix. And right in here is this intermembrane space. Now, the next two steps that we're going to get into are happening within the matrix. So that's Roman numeral 2 and 3 is in there. Okay, so the next step is called the transition, or also referred to as a Krebs prep. Where matrix of the mitochondria, the first step, remember, was to try to break down glucose. And now this will basically further break down. This time, it'll further break down pyruvate. If I slip and say pyruvate, it's really pyruvic acid. All right, so is there a black box for this as well? Yes. This is going to use another B vitamin. And that B vitamin is called CoA. So again, it's one of these B vitamins that are required for energy metabolism. All right, let's do a black box for that. So again, imaginary black box. It's really a few steps here. So in the transition, remember we had glucose was split to 2 pyruvates. Each pyruvate was 3 carbons each. 2 times the 3 gives you the 6, which is the original glucose. We're also going to enter in here the CoA, as well as 2 NAD pluses. So the thing that'll happen here is that the 2 pyruvates, you're going to break down each one. It's now going to turn into a 2-carbon chemical, and that's called acetyl. You're going to make 2 of these. So 2 2-carbon atoms, 2 times 2, now that's accounting for 4, right? So what happened to the other 2 carbons? Those other 2 carbons will be made for to make CO2. So 2 carbons here, that's 2, and then 4 from the 2 acetyl CoAs. What's going to happen to these 2 carbons? They're going to go out, and then those CO2s, you'll eventually exhale those, right? So at the alveoli, when you do that whole transfer mechanism, that happens there. Now, the acetyls, they will react with the 2 CoAs, and these guys together will combine, and you will form 2 acetyl CoAs. Remember, the acetyls are 2 carbons each, So you got 2 there, times 2 is 4, and then the other 2 was exhaled. Got that? NAD plus, on the other end, when this comes out, will capture some high energy from the pyruvate, and you're going to make 2 NADHs plus 2 H pluses. So again, we've scavenged some energy from the glucose, the original glucose, right? So we're going to tally those in a little bit. Okay, so that is a transition. Now let's go into something called the Krebs cycle. Krebs cycle is also in the matrix. Everything in the mitochondria will require oxygen. Okay, I'll reemphasize that at the very end. Remember, glycolysis, though, was in the cytoplasm, so that step is more of a oxygen-independent step. Okay, so the goal of the Krebs is to completely break down the carbon skeleton from the original glucose. And the other key goal, besides that, is that you're going to make the highest number of high energy carriers. Those high energy electron carriers, remember those are NAD, H, and FADH2. Okay. So let's do the Krebs. Before I do that, one last idea here is that, let's say each acetyl-CoA does the Krebs cycle one time. How many acetyl-CoAs do we end up making from the original glucose? 2, right? I'm going to summarize all the processes in that original glucose, and let's assume that we're talking now about the 2 acetyl-CoAs. So let's just do the Krebs cycle times 2. And that way we're accounting for the 2 acetyl-CoAs that originally came from the glucose that we started off with. So per glucose, you go through the Krebs 2 times. So let's say here, the 2 acetyls come in, 2 acetyl-CoAs. That's a 2 carbon compound each, right? Times 2, so 4 altogether. You're going to have 6 NAD pluses going in, 2 FADs, and then 2 ADPs, and 2 phosphates. All right. The acetyl-CoA will eventually all break down. How many CO2s can you make from 2 acetyl-CoAs? Altogether, 4. That will be exhaled. The 6 NAD pluses, I'm going to make the 6 NADHs plus 6 H pluses, 2 FADs, you're going to make 2 FADH2s, and then we'll also make 2 ATPs. Let's ask a question here. How many ATPs so far have been produced? If you look back at your notes, you should be getting the answer of 2 from the Krebs and then 2 from glycolysis. So 4 ATPs so far. You're like, that doesn't sound like 32 or 36. So where's the rest of the energy going to come from? The rest of the energy will come from all of these high energy intermediates. They still have energy, but you just haven't cashed in the energy yet. Okay? So look back at your notes and let's ask the similar question. How many NADHs are produced so far? And how many FADH2s? So this is actually the first time we've seen the FADH2. So altogether the net would be 2. And the NADHs, this one you have to look really more carefully. We made 2 in glycolysis and another 2 in the Krebs prep. We have 6 here. So 6, 2, and 2, that gives us 10. All right. Where does this energy now go to? Okay. So if you look at this diagram here, great majority of that energy will be made at the very end. All right. So let's recap some stuff. Glycolysis is that first phase. Take the glucose, split it, make the 2 pyruvates. You are going to use 1 ATP. That will destabilize the molecule, but also trap it. The net gain is going to be 2 pyruvates, 2 ATPs, and 2 NADHs. And it's the only one that's really happening in the cytoplasm. It is oxygen independent. The second phase is that transition. You're going to take that pyruvic acid and you're going to turn it into acetyl-CoA. Remember, the skeletal muscle can be done in low oxygen. You can regenerate NAD+, and that equation was the one that we did earlier when we went from pyruvic acid to lactic acid. Okay, remember that special enzyme was called a lactate dehydrogenase. All right, now, if you have the oxygen, you can complete the steps, and then we have this Krebs cycle, 2 acetyl-CoAs. Eventually, you're going to give off 2 molecules of CO2. Really should be 4 altogether. So 2 acetyl-CoAs, how many of these numbers would you get? It would be what you see right here. You're like, wait, that doesn't make sense. That seems less. Well, that would be from 1 acetyl-CoA. If it's 2 acetyl-CoAs, you would double all these. So it'd be 2 ATPs, 2 of those, and then 6 of that. This is Evie. She used to do a lot of gymnastics when she was young. And her big goal when she was small was to climb up this crazy rope that was like 2 stories high and ring the bell. Because if she got to do that, she got to get a free ice cream. And this girl is a carb lover. And look at how proud she was when she was like 5, 6 years. I think she was around 5. And I don't know how she figured out how to take a pose shot like that. It's kind of scary. Okay. All right, so we did our tally. The final step is this electron transport chain. So far, a lot of this is just tallies, right? What goes in, what comes out. Let's talk about the next part right here, which this part is more physiologic. So I want to break this down step by step. And when things are step by step, you know what that means, right? It may be a bolded question, and it turns out it is. So let's talk about the ETC, that electron transport chain. It is an aerobic step. It requires oxygen. Let's say also steps 2 and 3 need oxygen as well. However, the electron transport chain is the particular process where oxygen plays a direct role. You'll actually see it as part of these processes because we really haven't shown that yet. Okay. Now, to break this down a little bit more, let's blow up a mitochondria. And that mitochondria has an inner membrane And I'd like you to, when you draw your inner membrane, have it so that it kind of makes this, oh, what the heck. All right. Have it so that it has this kind of big slide in it. Whee, like that. All right, like so. Remember, this is the matrix. And this membrane is called the inner membrane, outer membrane, and then in between is that intermembrane space. All right. Now, a key part of this too is now we're going to focus in on these little guys here. These are called cytochromes. So the cytochromes are these blue dots. And the other big feature is this mushroom looking guy. And that mushroom guy is called the ATP synthase. I wonder what that protein does. We'll get into that. And really we're going to describe a process called chemiosmosis. It's kind of a weird term because it's not really using water, but it is using a concentration gradient. Now, what happens here is that, remember, you have these high energy intermediates. There was NADH and FADH2. What these guys will drop off are their hydrogen atoms. So, 1, NADH and FADH2 each release 2 hydrogen atoms. Remember, each hydrogen atom, what is a hydrogen atom made up of, right? It's made up of a proton, positive charge, and an electron. These hydrogen atoms will actually break apart into 2 electrons and 2 H pluses. Okay? Now, the electrons will get shuttled across the cytochromes as pairs. So step 2 is that the electron pairs are shuttled down the cytochromes. Remember what kind of process this was when something receives and then sends an electron? These are redox reactions. So this is a redox reaction that releases energy. The energy released is used to pump H plus into the intermembrane space. So when these hydrogen atoms break apart up here, the 2 electrons will be part of that, but then there's actually pumps along the membrane, and you will pump the H plus into this space. Notice what that does. It really creates an H plus concentration gradient. You're like, where did this energy come from? I don't really get it. Remember, these two electrons were shuttled by NADH and FADH2. Well, when did we make those? We made those in the breakdown processes of glucose. Remember, glucose was really high on the energy scale, and that high energy from glucose, why does it have high energy? It's because it has electrons that have a lot of energy. And then those electrons get shuttled to these next guys, NADH and FADH2. All right. So now you have this concentration gradient, right? The pH is dropping in that intermembrane space. That's not really a huge deal. But the H plus can diffuse through the ATP synthase. So H plus diffuses from a high concentration to a low concentration through the ATP synthase. That's that mushroom guy, right? When things go from a high to low concentration and the cells are put together this way, you can actually release energy. Energy is released. Okay, last couple steps here. The energy that is released here can be used or coupled to, that just means it's linked to, the production of ATP. They actually have figured out the fine details of this enzyme, this ATP synthase. They know how it's been studied a bunch. They know exactly how that ADP and the phosphates get slapped together when the hydrogen ions are leaking through. So energy released, used to combine ADP and phosphate to make ATP. Now, the electrons are shuttling, and I kind of made you purposely make this like little slide down here just to try to designate energy is getting lost. If you remember at the beginning of lecture, there is an atom here that can accept really low energy electrons because it doesn't care. It has such a strong attraction for electrons. It doesn't even matter if they're low energy. It will accept the electrons, and that atom is, of course, oxygen. Oxygen will receive the two electrons that now have low energy and combine with two H pluses. So after it has received those two electrons, it's now O with two electrons, right? Very reactive. It'll react with two H pluses, and now we make our other waste product, which it turns out is water. Or we make 6 waters at the end. This is the exact step where you actually make water in your metabolism. We actually make water as a waste product, which is pretty amazing. Now, this is really showing you the importance as well of oxygen. What if oxygen wasn't here at the very end to receive those low energy electrons? You guys agree that your cytochromes would actually back up? And if the cytochromes back up, then these intermediates don't have a way to release their electrons. And then these guys would back up. Well, remember, these intermediates are needed for things like the Krebs cycle, the transition step, and glycolysis. So if you don't have them as empty taxis, the whole process will actually shut down. So this is why when we talked about muscles, skeletal muscles specifically, that they can remake NAD+. For the rest of your tissues, when these hydrogen atoms are released, then you're back to the NAD plus and FAD, and these can return to those other phases of cellular respiration to then further break down glucose. And why is it oxygen dependent? You take this guy out, everything backs up. One of my jobs when I was in community college was one summer I worked for UPS. It was one of the hardest jobs ever, but I was making like $8.50, and back then in the 90s, I was making bank, all right? But super hard, 40-hour work weeks. At the end of my shift, I would take my shirt and literally wring it, and like there'd be sweat, dripping off of it, okay? But I was working not, in the brownie vans, but I was in the warehouses, and my job basically was to pull packages off of a conveyor belt and then build walls in a semi-truck. These semi-trucks would then be sent out to various cities to deliver these packages. So I was kind of the last man because I was like low on the totem pole. Why they give the last man like the new hire job? It was kind of a critical job, but I was basically like oxygen. Okay, now it all makes sense. If I were to not pull the packages off, these electrons, everything backs up. The conveyor belt would get way heavy. It'd slow down. The brownie trucks up here would not be able to drop off their packages, and everybody would be super angry at me, right? So I was kind of this low man here. But it was, now I know. I was like oxygen. I was really important back then. Okay. All right. So oxygen, kind of overall, really this drives the process. So without oxygen, steps really 2 through 4, the phases 2 through 4 would shut down. And if you really think about it, if this is not skeletal muscle, 1 would shut down too, right? Skeletal muscle can make ATP without oxygen, but really all the other tissues need to have oxygen. Where is oxygen more critical? Where is it actually playing a role in more? You'd say the mitochondria. The mitochondria is where oxygen is actually being used. And where is it directly used? That would be in the ETC, or the electron transport chain. All right. So that will wrap up the processes of cellular respiration. For the first few phases, 1, 2, and 3, focus on where it's happening, okay? But I would know what goes in and what comes out. Remember phase 1, there's 2 steps that we talked about. One was that first step that we actually add phosphate to the glucose. And the other step was the modified glycolysis that skeletal muscle can do because it has that specific enzyme. All right. And then step 4, which is the electron transport chain, I would go back and go through these processes, these steps one by one to see if they make sense, because this is one that I could possibly have you list as a short answer. All right. So the next lecture will finish off metabolism, and we will get into the breakdown of lipids as well as proteins. Great. Thanks, guys. If there's any announcements, I will put something up on Canvas.